Okay, here's the last part of chapter 11. So we're going to look at how the neurons, like put it all together and look at how the neurons actually work. The space that connects two neurons together, they're not actually touching. It's called a synapse, and that's where chemicals are being passed from one neuron to the other. So there's what we call the presynaptic neuron and the postsynaptic. The presynaptic is like neuron A, and then postsynaptic is neuron B. So the nerve impulse is going from A to B. That's what those two words mean. And one neuron could be both. One neuron could be A at one time and B at another time. Okay, so this would be the axon tail of the presynaptic neuron right here. And then it is joining to the next neuron, which is the postsynaptic. So you have tail joining to head, going to tail, joining to head, and that's the order that it goes on. And then you can have other nerves hooking up too. So you can see there's other ones coming in. So here's the axon, just like we saw, of the presynaptic neuron. The synapse is the gap in between. There's a space, and then postsynaptic would be here. Okay. You have different connections that you should write down and know the difference between these. The most common ones are axodendritic and axosomatic. So axodendritic is between the axon of one neuron and the dendrite of the other, or the axon of one neuron and the soma, the cell body of another. That's what we just saw. You can also have axoaxonal, which is tail to tail. You can have dendrodendritic, which is dendrite to dendrite, or somatodendritic, which is dendrite to soma, which is the cell body of the nerve. Okay, and there's two main types, chemical and electrical. Okay, so most common type of synapse, this is like the cookie cutter basic one that you always learn about, is the chemical kind that has neurotransmitters. So these are the chemicals that get passed between, like inside the synapse. At the very end of the axon on the presynaptic neuron, you have little synaptic vesicles, which are little sacs in the cell that are going to release neurotransmitter. Okay, and then you have a portion on the postsynaptic neuron that's going to receive those neurotransmitters. So one's going to pass it and the other one's going to receive it. Okay, so the transmission goes across the synaptic cleft from one neuron to the next. It's a chemical event, and that's what's going to cause the signal to pass, and this is unidirectional. It just goes in one single direction. So there's a few steps for this to happen. So first, the action potential gets to the end of the axon, and then you have voltage-gated calcium channels that will open, and calcium will come into the axon terminal. That's step one, steps 1 and 2. Then when the calcium comes into the presynaptic neuron, it's going to cause neurotransmitters to be released in vesicles. So the vesicles are going to erupt out and release the neurotransmitter. Then the neurotransmitter will diffuse across the synapse in like the space there, and then they're going to bind to an ion channel, and that's going to cause the channel to open. So this is a chemically gated channel. Then the neurotransmitter, we have to get rid of it somehow. So whatever was being passed, it could be dopamine, serotonin, melatonin, those are neurotransmitters. We have to get rid of it somehow, otherwise it'll keep stimulating that next that next neuron. So either you can have reuptake where the astrocytes are going to take it back up into the axon terminal. You can have degradation where an enzyme is going to come in and chew it up, get rid of the neurotransmitter, or it can just diffuse away somewhere else. It can just diffuse out of the way. But you don't want it to stay in that cleft, in that area, because it's going to keep stimulating the nerve. So here's what we're looking at. You have the action potential reaches the end of the axon. This is the presynaptic neuron. That's going to cause voltage-gated channels, calcium channels to open because the action potential is positive, right? It's changing the cell to positive. So these channels are going to open up once that hits positive. It's going to rush in, and that's going to cause these little vesicles that have neurotransmitters to erupt open and let out the neurotransmitters. They are going to bind to these gates, cause them to open, and cause the action potential to continue. And it'll ripple down the next cell. It takes a little while for the neurotransmitters to be released, diffuse, and bind, and that's like 0.3 to 5 milliseconds. So that's a little delay. So this step in the synapse is slower than when the nerve or the action potential is actually moving through. There's other types of chemical synapses too. There's electrically coupled ones where the cells, there isn't really a gap and the cells are right close to each other. That can be a lot faster communication. It can go both ways sometimes. So this is found like in eye movements, hippocampus, embryonic nerve tissue. So those are some exceptions, but most of them work like what we just saw. Okay, so postsynaptic potentials. So the neurotransmitter receptors cause graded potentials that vary in strength. It depends on how much neurotransmitter was released. This is also how drugs work. So if you have a lot of neurotransmitter, which can sometimes be drugs or they can do other mechanisms, it's going to have more of an effect. And then how long the neurotransmitter stays there. So if it doesn't get chewed up by the enzymes or reuptaken, it can cause more of a stimulus. So there are two different kinds of postsynaptic potentials. There's excitatory postsynaptic and inhibitory postsynaptic potentials. This is showing you one cell body, one soma, and a whole bunch of tails connected to it, and they can all be stimulating this neuron at the same time, and their stimulus can be added together. So some of these are going to be saying fire, like conduct a nerve impulse. Some of them are going to be saying the opposite, don't fire, and it adds them all together to determine what to do. So if these are hooked up, like let's say the green ones are saying yes, go, and the red ones are saying no, stop, it's going to add it all together, and then the neuron is going to determine should I conduct a nerve impulse or not. And it has to do with whether it's able to reach that threshold. Okay, so neurotransmitters bind to chemically gated channels, and like all of that's going to add together to determine if it's excitatory or inhibitory. Inhibitory is going to be like more of them are saying no. Excitatory are going to be more of them are saying yes. So it'll add it all together. So here's an example. Like this is one excitatory one that's saying fire, just one, but it's not enough to get it over that threshold. So it's not going to conduct a nerve impulse. I think there's another, this one's going to show inhibitory. So we'll see what that looks like. Yeah, so here, this one is an inhibitory response, and it's saying don't fire, and so it's not going to conduct a nerve impulse. And then let's look at the summation ones where several are talking to it. Some might be excitatory, some might be inhibitory. There's also temporal and spatial summation. So temporal is adding all of those action potentials up based on time, and then spatial is based on like where they are located. So here are two EPS, so these are two excitatory ones, but since they're too far apart in time, it's not going to reach the threshold. So it's like fire, minutes later, fire, it's not enough to reach the threshold because they have to be timed right. But if you have two of them that are close together in time, then it will reach fire, fire, fire. Then it'll reach the that level that it needs to. It'll get past the threshold. And then these are location. If they're hitting at the same place, look at this. You got them like spatially in the same place versus in the in different places. That's going to also cause it to sum together and cause an action potential. And then here's one that's excitatory, inhibitory, excitatory, and inhibitory together. Those are all going to add up and determine whether or not the nerve impulse is going to happen or not, or whether they cancel out. Okay, then you have synaptic potentiation. This is when if you use a synapse a lot, repeated use of synapse increases the ability of the presynaptic cell to excite the postsynaptic cell. So if you're using that synapse a lot, it's going to make it more usable and more likely to be excited. One of the reasons is because you're going to have more calcium hanging around at that terminal. Okay. presynaptic inhibition is when the excitatory neurotransmitter of one neuron is inhibited by another one. So sometimes they can do opposite things to each other, and if one is excited, it's going to inhibit the other one. Okay, so what do we have here? Location of event, cell body. So there's graded potentials. I'm not sure what this is showing. If it's a short distance versus a long distance, you could have a short distance graded potential and a long distance action potential that'll move down the entire neuron. So remember, the graded potentials are like short distance and they fizzle out. Okay, and then these are like factors that affect the action potential. Comparison of graded to action. And summation for graded. Summation doesn't occur with action potential. That's only with graded potential. I probably didn't say that. So you only see the excitatory and inhibitory with graded potentials, the weak ones. You don't see it with action potential. It's all or nothing. Okay, and then these are some of the channels that are involved in graded and action potential, and this is what they look like. Graded, action potential. Okay, so let's learn about some neurotransmitters. There's at least 50. You've definitely heard of some, even if you don't realize it. These are the chemicals that work in your brain that make your brain transmit action potentials. They are classified by chemical structure and function. So acetylcholine is the first one. This works with muscles. This is how your muscles contract. It was the first one identified. The abbreviation is ACH. Acetylcholine is how you say it. And it works with your nerves and muscles, so it goes between nerve and muscle, and it's degraded by this enzyme called acetylcholine esterase, ACHE. If your acetylcholine does not become degraded, it can cause your muscles to cramp, and that is a disease, as you would imagine anything. Any disorder of any of these mechanisms is a disease. You also have biogenic amines, so catecholamin. An example of that is dopamine, norepinephrine, or epinephrine. Those are made of amino acids of tyrosine. And then indolamines are like serotonin and histamine. Those are neurotransmitters. These are used in the brain, emotions, biological clock. If you have imbalances, that's associated with mental illness. And then amino acids are also neurotransmitters like glutamate, aspartate, glycine, and GABA. You might have heard of those. There's peptides like substance P. That's really important in pain. They just found that recently. Endorphins are neurotransmitters, and gut-brain peptides are neurotransmitters. And then purines, those are types of nucleic acids like ATP, adenosine, caffeine blocks adenosine receptors. So those are both neurotransmitters. And then you have gases and lipids. So gasotransmitters, nitric oxide, carbon monoxide, hydrogen sulfide. Those are all gasotransmitters. And this tells you what they do. Endocannabinoids like THC, they use the same receptor as THC, but these are not THC. They're G proteins. G protein linked receptors. I'll show you G protein stuff in a second. Okay, so neurotransmitters can have a lot of different functions. They can be grouped as their functions can be effects or actions. So effects, if they are, the effect could be excitatory versus inhibitory. So some are excitatory. They cause depolarization, which causes the membrane potential to go up, or inhibitory is hyperpolarizing and it makes it go down. So effect ones, it depends on which receptor they bind to. So GABA and glycine are inhibitory, glutamate is excitatory, acetylcholine and NE can have opposite effects depending on where they bind. So excitatory in skeletal muscles, inhibitory in cardiac muscles. Okay, actions, are they direct or indirect? Those are the two types of actions. So a direct action is the neurotransmitter binds to the ion channel and causes it to open. Or indirect, the neurotransmitter serves as an intracellular second messenger with G protein. So I'll show you that. And then you can have a neuromodulator. which doesn't cause excitatory or inhibitory, but it kind of turns up the volume, like it increases the strength of or decreases of the transmission. So it can influence synthesis, release, degradation, or reuptake of a neurotransmitter, can alter sensitivity of it, can be released as a paracrine, which is like where it acts on the same cell. And you also have the channel linked receptors. These are ligand gated ion channels. Their action is really fast, and it causes depolarization. So here is your channel when it doesn't have a ligand, and then here's your channel when it does have a ligand, and the ions can flow through. Okay, now G protein linked receptors. G proteins are really unique proteins that are inside the cell, just on the other side of the cell membrane. And these are really common ways that our cells communicate with each other, are through these G protein linked receptors. I'll show you a picture soon, but what happens is a neurotransmitter binds to it, it activates the G protein. The G protein changes shapes, and then it causes a ripple effect with these second messengers. They are like things like cyclic AMP, cyclic GMP, diacylglycerol, or calcium. And it's going to cause a signal to go into the cell and tell it to do something. That's what a G protein does. So here's a cute little thing. I think of this as like basketball, when you're like passing the ball to people to make a basket. So first the ligand lands on it, then the G protein changes in some way, and it gets passed to the next one and passed to the next one, and then it's going to bind to that, and then it causes this, and then it causes this enzyme to activate, or something, or this ion channel to open, or it's going to talk to the nucleus and tell it to turn on certain genes. Something will happen. Neural integration is when the neurons are functioning together in groups. You have tons of neurons lumped together, like strands of hair, and they work together to do a task. They integrate. Neuronal pool is a whole bunch of functional groups of neurons, so they can work together in that way. So simple one, you have a single presynaptic fiber. It branches out to several other neurons. You can have a discharge zone, where the neurons closer to the incoming fiber generate the impulse, or a facilitated zone where the further away neurons don't get as excited, but they can also grab the signal. So here's your presynaptic fiber. Right in the line, right front and center is the discharge zone, and then the facilitated zones are a little bit further away. They're kind of peripheral. Serial processing, the input goes through one pathway in an all or nothing fashion, so like a spinal reflex. Sometimes these don't even make it to the brain. They just stay in the spinal cord. So reflexes are rapid automatic responses to stimuli. When the doctor uses the hammer on your knee, that's a reflex. There's what we call a reflex arc, where the receptor receives the information, it goes to a sensory neuron, then it goes to an integration center, usually in the spinal cord. Then it goes to a motor neuron and then an effector, and you kick your leg. So here that is receptor. This is the spinal cord. This is an interneuron. The blue is a sensory neuron, and the red is a motor neuron, and then it's going to cause a response. So that's a simple reflex arc. Parallel processing is where there's several pathways moving together. This is higher level mental functioning, like you smell something and it reminds you of another experience. That would be parallel processing. There's 4 types of circuits. There's diverging, converging, reverberating, and parallel after discharge. So this is a diverging one, where it starts from one and then it spreads out to many. So a single neuron can activate different motor neurons. Converging is where these 3 inputs are going to converge into one and cause one output. So different sensory stimuli can elicit the same memory. Like if you smell something, taste something, hear something, it might cause you to think of the same memory. And then reverberating is when they kind of loop back and recycle the impulse. This is involved in breathing, sleep-wake cycle, motor activities like walking. And then parallel after discharge So this is where you have a whole bunch that are together stimulating that output. So this is like mathematical calculations or something. Okay, when you are developing, the nervous system starts as a neural tube, and it starts from ectoderm. I think we've talked about this earlier. So you have these special cells, neuroepithelial cells, and that's what's going to become the brain and spinal cord and neurons. There's a growth cone that is at the tip of the axon, and that's going to start to grow out. You have nerve growth factor that keeps those cells alive and allows it to grow out. And then the axon is going to grow till it finds its target and form a synapse between it and another neuron. Remember those glial cells that help support? Here's one of those. The astrocytes is going to provide support so that these neurons can grow, right? Before you are born, about 2/3 of neurons die because they're fishing out, trying to find connections, and they can't quite find them. So they're going to undergo apoptosis. So your brain is really trying to figure itself out and grow, especially in young children, and we need to make sure that it has good opportunities to connect to the right neurons. During childhood and adolescence, you're growing tons of neurons, you're pruning some away, you're doing apoptosis. If you have excessive pruning, that could lead to like schizophrenia, so that's something they can look at. Neurons are amitotic after birth. That means they do not do mitosis after birth, but some of them do divide. Like they used to think that no nerve cells divided, and if they died, they died. But now they know that some are dividing. Okay, so that's a neuronal growth cone where it's trying to find a synapse to hook up to. And that's the end.